Our DNA is like a library found in the nucleus of our cells, with thousands of books. Some of these books, called genes, are extremely important because they carry the recipes for every single protein found in the cell. These genes are scattered among 23 pairs of chromosomes, which are like the bookcases of the library. Chromosomes come in homologous pairs because one comes from mom and one comes from dad. Each chromosome of the pair carries different versions of the same genes, called alleles. Now, on the molecular level, DNA is made up of 2 strands of nucleotides, so each gene is just a segment of this nucleotide sequence. There are 4 types of nucleotides: adenine, guanine, thymine, and cytosine, or A, G, T, and C. Gene expression includes transcription and translation. Transcription is where the enzyme RNA polymerase uses the gene as a template to create a molecule that can leave the nucleus. This molecule is called messenger RNA, or mRNA, and it has the same nucleotide sequence as the gene, with one tweak. It has uracil nucleotides, or U, instead of thymine. This mRNA molecule, or message, is encoded so that any 3 nucleotides equate to a specific codon, which codes for an amino acid, or is a stop codon, which signals that the protein is complete. In translation, specialized proteins in the cytoplasm, called ribosomes, use the mRNA template to recognize the specific codons, and match them with the corresponding amino acids that'll make up the protein. Now, there are 64 different codons, and each of them codes for a single amino acid, but there are only 20 amino acids. That's because some amino acids are encoded by more than one nucleotide triplet. Now, a mutation, put simply, is an alteration in the nucleotide sequence of one or more genes, but can sometimes affect large chunks of chromosomes. These mutations can affect the chromosomes in somatic cells, meaning any cell in our body other than the gametes, or the chromosomes in the gametes. Mutations in gametes are called germline mutations, because they can be passed on to the next generation. Now, mutations can happen spontaneously, or they can be induced by mutagens. Mutagens include physical agents like UV rays and chemicals, to biological agents like viruses. Often mutations happen during DNA replication, which happens right before a cell divides. Let's start with small mutations, involving the nucleotide sequence of a single gene. 3 common types include substitutions, when a nucleotide is swapped or substituted for a different one, like swapping U for A. Deletions, when one or more nucleotides are deleted, like deleting U, for example. Or insertions, when one or more nucleotides are added, like adding A into the sequence. With substitutions, the result depends on whether that swap results in a new amino acid. And if it did, what matters is how that new amino acid affects the overall folding and function of the protein. For example, let's take the codon UGU, which codes for the amino acid cysteine. A point mutation in the last U for a C results in the codon UGC, which also codes for cysteine. So in this case, the resulting protein isn't changed at all, and this mutation does not have a functional consequence. So it's called a silent mutation. Now instead, let's say that in the UGU codon, there was a point mutation in the last U for an A. That results in the codon UGA, which is a stop codon. A stop codon makes the ribosome stop building the protein. And this kind of mutation is called a nonsense mutation, because it results in a much shorter protein that can't function properly. Now let's say that in the UGU codon, there was a point mutation in the G for an A. That results in the codon UAU, which codes for the amino acid tyrosine. This is called a missense mutation. Depending on which amino acid it codes for, missense mutations can be conservative or non-conservative. Conservative means that the resulting protein can still function properly, because the switch coded for an amino acid with similar chemical properties to the original one. In this case, both cysteine and tyrosine are polar amino acids, so the protein can still function pretty well. Kind of like sweetening lemonade with honey instead of sugar. But now let's say that in the UGU codon, there was a point mutation in the last U for a G. That results in the codon UGG, which codes for the amino acid tryptophan, a nonpolar amino acid. The resulting protein cannot function properly. Kind of like trying to sweeten lemonade with salt. Not gonna work. This is what happens in sickle cell disease. The hemoglobin protein has a mutation that changes the amino acid glutamate, which is hydrophilic, for valine, which is hydrophobic. And the resulting hemoglobin protein is more frail, which makes it hard for red blood cells to carry as much oxygen. Then there are the insertions and deletions. If these mutations are in multiples of 3 nucleotides, then they'll displace the reading frame of the mRNA codons by exactly 1 entire codon. For example, let's say that you add 3 or 6 nucleotides, or take away 3 or 6 nucleotides. That means that the majority of the protein will have the same amino acids, with only a few added or taken away. And that's called a non-frameshift mutation. If the deletions or insertions are not multiples of 3, like adding 1, 2, 4, or 5 nucleotides, or taking away 1, 2, 4, or 5 nucleotides, then it's called a frameshift mutation, because it means that the reading frame is shifted. Basically, every single codon after the insertion or deletion will be off, and the entire protein will be made of completely different amino acids. For example, the mRNA sequence UCU, CCA, GCU codes for the sequence of amino acids serine, proline, alanine. If we add a U in the third place, the resulting sequence is UCU, UCC, AGC, U, which codes for the amino acid sequence serine, serine, serine. So the amino acid sequence, and in turn, the resulting protein, is severely compromised and non-functional. Now let's switch gears and look at large-scale mutations, which includes having an abnormal number of chromosomes, or chromosomes that are structurally abnormal. These mutations mostly arise because of errors during the formation of gametes, so they're often associated with genetic disorders. First, let's look at an abnormal number of chromosomes. When the gametes are forming, if something goes wrong, they can end up with additional chromosomes or missing chromosomes. We call this aneuploidy, and depending on which chromosomes we have more or less of, aneuploidy can give rise to genetic disorders, like Turner syndrome, when the person only has one X chromosome instead of 2 X chromosomes, or an X and a Y chromosome. Or Down syndrome, when there's an extra chromosome 21, so 3 in total, which is why it's also called trisomy 21. Another kind of number abnormality is polyploidy, which is when a gamete starts out with an extra set of chromosomes. For example, an egg might have an extra set of chromosomes, so 2 sets or 46 chromosomes in total, and a sperm might have a single set of chromosomes, so 23 chromosomes. When they meet, the result would be triploidy, meaning that the resulting zygote has 3 sets of chromosomes, or 69 in total. If there's tetraploidy, it means that there are 4 sets, or 92 chromosomes in total. Triploidy and tetraploidy are examples of polyploidy, and they're not compatible with life. Blech. And finally, let's look at structural abnormalities in chromosomes, like a deletion, duplication, inversion, or translocation. These can happen because of errors in gamete formation or because of mutagens like radiation. Deletions are pretty straightforward. Basically, a whole chunk of the chromosome goes missing. An example is a syndrome called Cri-du-Chat, or 5p- syndrome, which is when there's a deletion on the short arm of chromosome 5. Then there are duplications, which can happen when a gamete ends up with an extra chunk of chromosome that might be broken off from another chromosome. That extra piece often attaches to the homologous chromosome, which now has duplicate chromosomal segments that code for the same genes. Next are inversions, which are when a chromosome breaks off and reattaches to the same chromosome, but it gets flipped around in the process. So if the original order of the genes in the chromosome was ABCD, now it's ABDC. And finally, there are translocations, which is when a part of one chromosome breaks off and is exchanged for a part of another non-homologous chromosome, which might result in a fusion between genes that were initially separated. A classic example is the Philadelphia chromosome, which is a chromosome 22 with a bit of chromosome 9 on it. Right where they come together, there's a fusion between the BCR gene, initially in chromosome 22, and the ABL gene, originally in chromosome 9. So there's a fusion gene ABL-BCR, which leads to uncontrolled cell division that results in a type of leukemia. In general, it's easy to think of DNA mutations as always causing problems, but some mutations can help the species as a whole survive because they lead to diversity. Without them, we wouldn't have any genetic diversity, and we wouldn't have the ability to evolve over time. In fact, all of the uniqueness that makes individuals different from one another can be traced back to a genetic mutation of some sort. Alright, as a quick recap. Mutations are alterations in DNA. Small-scale mutations include substitutions, insertions, and deletions. Substitutions do not alter the reading frame of mRNA codons, whereas insertions and deletions of a number of nucleotides that is not a multiple of 3 result in frameshift mutations. Large-scale mutations involve abnormalities in chromosome number or structure. Numerical abnormalities are aneuploidy, which refers to one or more chromosomes extra or less, and polyploidy, which refers to entire sets of extra chromosomes. Blech. Structural abnormalities include translocation, inversion, duplication, and deletion.